Students, in this tutorial, Molecular Basis of Inheritance Part 5, we'll be looking at the concepts genetic code, and we'll also be talking a little about mutations. So in the first slide, we are going to talk a little about replication and transcription. These are two processes that you have already studied, and by now you know that in both these processes, what we're doing is we're taking one nucleic acid and we're making another nucleic acid by following the complementarity rule that was given by Irwin Chargaff. So in case of replication, what we do, we take a DNA, and if that DNA template has an adenine in the newly formed daughter DNA, it will have a thymine. So this will tell us exactly what are the sequence of bases that should come on the new daughter DNA strand. The complementarity rule will tell us the sequence. Similarly, in transcription, you're using a DNA to make a RNA. So wherever there is an adenine on the new RNA strand, you will be pairing it with uracil. Or if there is a guanine on the DNA template, you will be uh, pairing it with cytosine. Okay, so it is fairly easy to conceptualize because here the only thing that's happening is you have to copy one nucleic acid into another nucleic acid. In translation, however, there is a problem because you are taking or transferring information from one biomolecule into another biomolecule. So the biomolecule in question here is the nucleic acid, which is a polymer of nucleotides, and information from here has to be transferred to the biomolecule, which is a protein, which is a sequence of amino acids. And not helping the cause is that there is no complementarity rule that exists, okay, to determine what sequence of amino acids should come in your newly formed protein. So this is a question that baffled a lot of scientists at that time. They didn't know exactly what was dictating the sequence of amino acids that should come in the newly formed protein. Okay, so in other words, they wanted to know exactly how the cell produced a protein. Okay, now the idea came from experiments that showed them that if you change the nucleotides in nucleic acids, okay, there will be a corresponding obvious change in the amino acids in the protein that is coded by that nucleic acid. So this kind of information they had through experiments. That is, any change in nucleic acids would result in a change in the amino acid sequence in proteins. So this kind of gave rise to the idea, okay, of a code being present that would dictate the amino acid sequence. Okay, so this led to the proposition of genetic code, and uh, they they knew that there is a code now, but still the deciphering of the code was very challenging, and people struggled with it for years. Now, in your book, they kind of summarize the history of how the genetic code was cracked, okay? And they just give you a few tidbits and a few names here and there. But the history, if you really delve into it, okay, and you want to learn about it and understand it more, is very interesting. And I have linked some videos in Google Classroom, which makes this more clearer. Okay, it gives more clarity to the whole process of how the genetic code was deciphered over a number of years, and how there were people working in different countries, to put all this together. So in your book, they've mentioned the prominent names, okay, of some men who really did, whose work was paramount in finding out and cracking the genetic code. The first name that is given in your book is George Gamow. So somewhere in the year 1953, just to summarize his work, he proposed. Okay, he didn't do actual experiments, but he proposed that there is a set of three bases that could form a code that would encode for the 20 standard amino acids that occur in nature. And these 20 standard amino acids, which were known to occur in nature, were the building block of proteins. So George Gamow just planted that idea that the code is not of one base but a combination of three bases. But the actual experimental evidence and the proof that the cracking of the code that we now know is definitely that the code is made of three bases was done by Marshall Nirenberg and one of his PhD students by the name Heinrich Mattei. So these men were the ones to do a series of precise and logical experiments and crack the code. So these people were working in the US. Okay, and you also had another scientist that's mentioned in your book, and he's of Indian origin. That means he was born in India, but later on he acquired US citizenship. He is Har Gobind Khorana. He also made important contributions that led to the deciphering of the genetic code, and therefore he is given credit. And in the year 1968, Marshall Nirenberg shared the Nobel Prize with Har Gobind Khorana for this work of deciphering the genetic code. A name mentioned in your text is Severo Ochoa. He is also again a Spanish born American scientist because later he migrated to America and acquired citizenship there. His work also influenced the deciphering of the code, and what he did was he kind of found out different enzymes that could synthesize RNA. And one such enzyme which is capable of synthesizing RNA is polynucleotide phosphorylase enzyme. So all these different people, they worked on small little bits, and finally the whole code was deciphered. So the contribution is really, you can say, an outcome of all their efforts. Now, giving you a quick look at the genetic code. Okay, so this was finally the code and the chart that they made to understand, okay, which code, or now they call it a codon, coded for a particular amino acid. Now, the same code is very simply given in your text in table 6.1. So, what I want you to do is take a look at table 6.1, okay, and find out which code for which amino acid. Now if you look here, okay, you can see here it is giving you the code, and here it's giving you the amino acid names, and this outer circle is giving you the short forms for the different amino acids. Now please understand that you're not supposed to know all the codons and which subsequent amino acid they are coding for. You're not supposed to know all the codons and all the codes for the different amino acids, but you just need to know the start codon and the stop codon. So we're coming to that. Okay, so going back to what is a genetic code? If you are asked a simple question, what is a genetic code? The genetic code is nothing but the sequence of bases in the messenger RNA that code for a particular amino acid in the protein synthesis. So this is the definition given in your book. Okay, obviously the mRNA is taking the code from the DNA. So genetic code is actually the code on the DNA which is copied into the RNA and used to make the protein. Okay, now the code is made of three nucleotides, and thus it is called as a triplet codon. Now some books quite simply call it the codon, but always keep in mind it's made of three nucleotides, or you can say three bases, and therefore it's called as a triplet codon. So you can see an example here, U. Okay, you can see a sequence of three nitrogen bases, and this is the codon for the amino acid phenylalanine. Similarly, you can see this codon UUA, it is coding for leucine. Okay, now take a look. This is very interesting. You have UUA coding for leucine, and you have another codon for leucine, which is UUG. Similarly, you can see here for phenylalanine, you have UUC also coding for the same amino acid. So keep in mind that although it is one code for one amino acid, okay, you can have two amino acids coded by sorry, one code for one amino acid. You can have the same amino acid coded by different codons. So you can see here, same amino acid coded by different codons. So this is to be kept in mind. Okay, coming back to the genetic code, just to show you how exactly you can find out which codon for which amino acid here. If you take a look at this, okay, phenylalanine. Now you want to find the code for this amino acid phenylalanine. What is the codon? So you will simply look at the inner circle, the middle circle, and the outer circle. So the code is T T T or T T C. So there are two codons for the same amino acid of phenylalanine. PHE is the symbol or the short form for phenylalanine. Now in your book, if you notice the first block of the table, it's given as UUU. Clear? Because it's given in terms of RNA, whereas here it is in terms of the code on the DNA. So don't get confused with that. So either you can say TTT codes for phenylalanine or UUU codes for phenylalanine. It is understood. Okay. Similarly, we'll take leucine. So leucine, this is your short form LU. Now look at this. You have TTG, which is coding for leucine, and you have also TTA coding for leucine. So you have two different codons for the same amino acid. So this is important now. What are the features finally of the genetic code? And we call them as the salient features of the genetic code to be kept in mind. Okay, so the first point is that it is a triplet. There are 64 codons that can be worked out because it's a sequence of 3 bases. So if you combine these 3 bases in 4 bases, adenine, guanine, cytosine, and thymine. These are your 4 nitrogen bases. These 4 bases, if you combine them in different ways into threes, you will get 64 combinations, out of which 61 are actually coding for amino acids and 3 do not code for any amino acid, and therefore they're called as stop codons or nonsense codons. So you got to know which are the stop codons. These are UGA, UAA, and UAG. Okay, so these are your stop codons. Try and write it in terms of uracil and not thymine. Clear? Then one codon codes for only one amino acid. This I showed you, right? So one codon, one amino acid. You will never have one codon coding for two different amino acids. So therefore we call the genetic code as unambiguous because one codon codes for only one amino acid. But the code is also said to be degenerate or redundant. In some books, this word is given as redundant because you can have some amino acids coded by more than one codons. So I gave you the example of phenylalanine, leucine, and here you can take a look at proline. You have the same amino acid, two different codes. Clear? So this is why we call the code as degenerate. So many a times you get questions like this. Why is the code called degenerate? Why is the code called unambiguous? So you should be able to give these reasons. Then the next salient feature is that the code has to be read in a continuous fashion. There are no punctuations, so you cannot say U dot G dot A or U comma G dot comma A. Okay, it has to be read in a continuous fashion. The next point, fifth point, it is universal. So if you take a bacteria or a human being, if it is the codon UUU, it is coding for the same amino acid, that is phenylalanine. This is not it's a general rule. Okay, so this is not like a sure shot rule because you can have some exceptions and you will find them in mitochondrial codons and in some protozoans, where the code may differ in case of the protozoan and the human, or the protozoan may be slightly different than other animals. Uh, AUG, which is the start codon, again you got to know this start codon AUG has dual functions, meaning it is doing two jobs. It is coding for methionine amino acid, and it also acts as the initiator codon in case of translation, the start codon. So it's also called as start codon. So please keep these salient features in mind. Now, so this was regarding the genetic code. Coming to the second concept for today, genetic mutations. So we're going to talk a very little about it because this is covered in detail if you remember in chapter 5. Okay, so we've already touched upon point mutation. This is just a kind of uh, a little, you can say, connection to that chapter. So you have point mutations. Point mutations, we know a classic example, sickle cell anemia, where the mutation is taking place in a single base pair. So in sickle cell anemia, we have learned in great detail that instead of GAG, a single base changes, that is from A, it changes to U at the sixth position of the beta globin chain of hemoglobin, as a result of which the amino acid itself changes. So instead of having glutamate in the normal hemoglobin chain, you end up getting valine, as a result of which there'll be sickling of the RBC and all the physiological effects that follow. Okay, so this is an example of point mutation, wherein you can see something going wrong in the genetic code. Frame shift mutation is when there is a base that is either inserted or deleted, which causes a shift in the reading frame. And why I focus on frame shift mutations a lot is that it is apparent that these are the causes of several genetic diseases, such as Tay-Sachs disease and certain cancers, all are attributed to such mutations taking place in the DNA of individuals. Okay, so this frame shift mutation and understanding of this does help a lot in studying genetic diseases and disorders. In your syllabus, of course, we don't have any disorders listed in frame shift mutation. You just have to know the definition and how it works. Okay, so just to give an example of how frame shift mutation works. Suppose you take a sentence in English, Ram has a red cap. You can see here now that Ram has a red cap. I have decided to introduce the letter B. Okay, so if you notice, I've taken each word to have three letters. So it is representative of your codons. Okay, so Ram has, or you can forget just to make sense, red and cap. Now see what happens to the word red. If you decide to insert a letter B, from this point onwards, the reading frame of the codons change. You're supposed to read codons in sequences of three. Okay, so an insertion of one letter is causing the reading frame to change. Now think in terms of your RNA. So suppose you have UUU. This is a codon for phenylalanine. We all know by now. Suppose you end up putting a C that comes here. Okay, you're inserting a base. You're adding a base. The codon now changes to CUU, which is a different amino acid. It is leucine. Okay, and you can see this U gets added to the next codon, so this codon will change. So you can see this happening when a base is added, mutation is being brought about. Same thing could happen if you omit. Okay, so if you omit also, as you can see that's happening in the sentences here, it will change the order of the reading frame, causing disruption. So the sentence from here on does not make any meaning. Similarly, your DNA if there is an insertion or deletion of a base from that point onwards, it does not make any sense. Okay, however, notice this thing. Suppose an entire alphabet is added. Okay, sorry, entire word, big. So it's only at this point that there is a change, whereas the rest of the sentence remains the same. Same thing happens with codon. So if you put an entire codon, that means three or more bases are added, three or multiples of three are added. It does not have such a big difference on the reading frame. Okay, so remember these 2 points. Insertion or deletion of one or two bases alters the reading frame from that point onwards. Insertion or deletion of three or more of its multiple bases will keep the reading frame unaltered from that point of insertion or deletion. Okay, so this is about frame shift mutations, and we've talked a little about point mutations. Clear? So with this, we end part 5. I have dealt with the concepts of genetic code and mutations in this tutorial. Thank you.